So I did my project on amyotrophic lateral sclerosis, ALS. So ALS is a specific neurodegenerative disease. It leads to the death of neurons that control voluntary muscles. It really became widely known in the US after famous baseball player Lou Gehrig, who was on the cover slide, was diagnosed in 1939. It's age-related in that it has a late mean age of onset, usually after 50. Epidemiology of the disease. The incidence globally within a population of 100,000 people, there are 2 new cases of ALS per year globally. Within the US, the incidence is 1.5 person years. Prevalence, globally, the prevalence of ALS is 0.86 to 2.5 cases a population of 100,000. Within the US, the prevalence is 5 cases per population of 100,000. Mortality, globally, there are 2 deaths per 100,000 people per year from ALS. Within the US, it's 1.5 deaths per 100,000 people per year. This is a bar graph showing prevalence rates in different age groups for the years 2012 and 2013. So you can see after age 50, there's a spike in the prevalence with the peak prevalence in the 70s, and then it kind of goes down after the 70s. These are prevalence rates based on sex, again for the years 2012 and 2013. There's a slightly higher prevalence rate for men versus women, and this is shown. The etiology, the origins of ALS really remain unknown, but it is generally associated with the degeneration of cortical and spinal motor neurons. The disease is categorized in 2 forms. The most common is the sporadic one. 90 to 95% of cases arise from it, and it lacks a genetically inherited component. Least common is FALS, familial ALS, with an associated genetic dominant inheritance factor. Early signs of ALS include difficulty walking, trips and falls, trouble maintaining posture, slurred speech, muscle cramps. Really, the degeneration of the voluntary muscles. You start to see that. As the disease progresses, there are greater complications, which include breathing problems. When you have the breathing muscles start to become paralyzed, you have breathing problems, and the most common cause of death for ALS patients is respiratory failure. Speaking problems get worse. Eating problems can lead to a feeding tube and a greater risk for aspiration pneumonia because you're swallowing, the muscles that that control swallowing become damaged. So there's a greater risk of food, liquid, saliva getting into your lungs and causing an infection. Dementia is also present in later stages of ALS. These are 2 major complications. The first being intervention via mechanical ventilator. The individual on the left in the wheelchair, he's on a ventilator because his breathing muscles have atrophied to a point where he can't breathe on his own. And as I mentioned, aspiration pneumonia, the X-ray on the right, is a great risk, whether it's in ventilator ALS patients or just non-ventilator ALS patients. You have a general weakening of your swallowing muscles that puts you at a greater risk of aspiration. Diagnosis is tough. ALS is a tough disease to diagnose. There are a lot of muscular and nerve disorders and other neurodegenerative diseases, of course, that mimic its symptoms. So there is no single golden standard test or examination for diagnosis. there's only a series of diagnostic tests. The most common examinations include electromyogram, EMG, where you're looking at electrical activity in the nerves, top right image. And the next is nerve conduction study. Again, you're looking at the health of the nerves. And standard MRI to really rule out any other things that could be leading to your symptoms, such as a tumor in your spine. For final diagnosis, neurologists would incorporate all the results of these tests and the clinical examination. And if these test results aren't that salient, they don't clearly point to ALS, a decision to retest the individual can be made after a certain period of time, a couple months, because the individual might be in the very early onset of ALS. Going into the molecular and cellular mechanisms, ALS is defined by the death of upper motor neurons, UMNs, in the motor region of the cerebral cortex and the brainstem. It's also marked by the death of the lower motor neurons, LMNs, in the spinal cord. These neurons control voluntary movement. So when you have the death of these neurons, you're going to have atrophy of skeletal voluntary muscles around the body. And at the bottom right, you can see an image of the muscles, and you're going to have atrophy everywhere. It's going to have it's going to lead to all the symptoms that we talked about. So the primary motor cortex is shown here. That's where the upper motor neurons are. Also in the brainstem here, and the lower motor neurons of the spine. Going deeper, going into more detail, the upper motor neurons, they originate in the motor cortex or brainstem, but they descend into the spinal cord to the appropriate spinal nerve root, where they synapse with the LMNs. So nerve signals carried by UMNs continue onto the muscles, but only via the lower motor neurons. Lower motor neurons of the spine are found specifically in the anterior gray column or the anterior nerve roots of the spine. They function by innervating the fibers of skeletal muscles and thus causing their contraction. They are what maintains the link between upper motor neurons and being able to control muscle voluntary movement. The most commonly identified mechanism behind ALS is the mutation of a gene that encodes the antioxidant enzyme SOD1, which is superoxide dismutase 1. This mutation leads to structural instability and misfolding in this enzyme, which can lead to the aggregation of motor neurons in the central nervous system. But the mechanism of action of SOD1 has several major hypotheses that we'll go through. The first being glutamate excitotoxicity. Studies have found that the spinal cord and motor cortex of ALS patients and mouse models have reduced astroglial glutamate transporter, known as EAAT2. During the healthy neurotransmission process, glutamate that's released into synaptic cleft activates postsynaptic receptors. But after the release of the synaptic vesicle, EAAT2, or the excitatory amino acid transporters, they remove that glutamate from the synaptic cleft. So reduced EAAT2 in studies has shown to increase extracellular glutamate that accumulates. And this can lead to overstimulation of glutamate receptors and excitotoxic neuronal degradation. So both in sporadic and familial cases of ALS, this glutamate excitotoxicity leads to neurodegeneration, and specifically through the influx of calcium and sodium ions and activation of the calcium-dependent enzymatic pathways, or really by the generation of free radicals, because remember, SOD1 is an antioxidant. So another proposed mechanism is the functional and structural abnormalities in the mitochondria. In ALS patients, mutant SOD1, the misfolded type, is deposited on the cytoplasmic surface of the outer membrane and matrix of the mitochondria. This is considered the reason for mitochondrial dysfunction. And this dysfunction leads to a whole host of problems, which include abnormal functioning of ATP production, calcium homeostasis is disturbed, axonal transport of mitochondria is disturbed, and the triggering of apoptosis. All of these have been found in ALS patients. They have found to occur, and they all contribute to neuronal degeneration. This is just a graphic conveying what I just talked about. Another proposed mechanism is impaired axonal structure and transport defects. Axonal transport has been shown to be defective in ALS patients. In mouse models with mutant SOD1 mice, both anterogade and retrograde movements transport was impaired. Several pathways are possible for this. They can include defective mitochondrial function, which we just talked about, or disruption of kinesin function by tumor necrosis factor, or excitotoxic damage by glutamate, which we talked about first. Damaged axonal transport, it leads to a collection of autophagosomes, mitochondria, neurofilaments in degenerated motor neurons, which leads to their loss. Free radical mediated oxidative class. We've addressed this topic many times in the class. So SOD1 is a key antioxidant protein. Cytotoxicity can be induced by the mutant SOD1, which leads to ROS accumulation, oxidative stress. So studies have actually found oxidative stress, free radical accumulation, and oxidative damage in CSF, cerebrospinal fluid, serum, and urine samples of ALS patients. So it definitely is occurring. So genetics, pathological presentation of familial and sporadic ALS is very similar. But familial ALS occurs due to specific gene mutations, gene loci mutations. Besides the SOD1 mutation, there have been around 18 other gene mutations that have been identified, such as TRARDP, FUS, and ANG, that have been implicated to be in association with familial ALS. Totally, around 50 to 60% of FALS cases have been attributed to specific gene loci mutations. Current therapeutic approaches. There is only one FDA-approved drug that slightly enhances prognosis of ALS patients. This is the only proven treatment. It's called Riluzole, shown in the bottom left. It comes in the form of tablets. And what it generally does, it works to decrease the neurotoxic effect of glutamate. The other alternative besides Riluzole for treating ALS is really good palliative care to only treat the symptoms. You can try to mitigate the symptoms by specialized care for ALS patients, and this can really boost quality of life. Going into the specific mechanism Riluzole. So Riluzole is known to induce presynaptic inhibition. Glutamate is subsequently released from the cerebrocortical nerve terminals, and it the drug inactivates voltage-gated sodium channels and functions as an NMDA receptor antagonist, a non-competitive one, that is. And the net result is mitigation of neurotoxic effects of glutamate. On the bottom left is the skeletal structure of glutamate, and on the right is a graphic representation of this mechanism. There have been some indications of future directions for researchers in studying treatment of ALS. Studies have established similarities between SOD1-associated ALS and Down syndrome etiology. Both really involve hyperphosphorylation of tau, and future investigation into their relation may provide some novel treatment methods for patients. Additionally, investigating various shared environmental genetic susceptibilities between neurological disorders like Parkinson's disease and just general frontotemporal degeneration and ALS, as well as monitoring identical treatments administered to patients of these neurological disorders, can result in some breakthroughs. It's pretty promising. The featured scientist I chose is Dr. Nazem Atassi. He is the Associate Director of the Neurological Clinical Research Institute at Mass General Hospital. He's also an Associate Professor of Neurology at Harvard Medical School. The research focus of his lab group is translational research and clinical trials in amyotrophic lateral sclerosis, ALS, and other neurodegenerative disorders. A recent publication of his, glial activation, it's titled Glial Activation Co-Localizes with Structural Abnormalities in Amyotrophic Lateral Sclerosis, came out in 2016. And just briefly going into the purpose of it and its findings, Dr. Atassi went in with the experimental objectives of examining brain morphological irregularities in association with the typical activation that is seen in ALS of glial cells, which are the non-neuronal support cells. And he used an investigational PET radioligand to bind to activated glial cells to really look at the changes in the cells and function as a marker of activation-induced inflammation, which is seen in ALS. A cohort of 10 ALS patients and another cohort of 10 matched healthy controls were put through the PET imaging, but also diffusion tensor imaging, which is known as DTI, using MRI, magnetic resonance, in the study. The purpose of the DTI imaging was to record indices of spatial statistics, tractography, fractional anistropy, and diffusivities in various ROIs, regions of interest. Coupled with the collected measures of cortical thickness, which are actually measured in a different way from surface analysis, these metrics all conveyed any morphological change in the brain. So having compared these potential changes in morphology and glial activation, which was measured by the radioligand uptake using PET, following the study, Dr. Atassi's findings conveyed that increased uptake of the radioligand correlates with changes in fractional anistropy, which is a measure using diffusion MRI imaging, like I mentioned, and cortical thickness. So in essence, his study is establishing a link between specific mechanisms of ALS pathogenesis, which are inflammation, gliolysis, which is the reactive change of glial cells in response to damage to CNS, the central nervous system. So there's a link between this and ALS brain morphological changes, of which the most salient detectable in this study included cortical thinning and gray-white matter changes, which were detected by the DTI. One figure that was very comprehensive and supported Dr. Artasi's work was figure 1. Really what figure 1, which is on the left, is demonstrating how the radioligand, which is known as 11C PBR28, the PET radioligand, its uptake correlates with decreased fractional anistropy in the brain's motor cortex. So that marks the progression of ALS. Another great figure, figure 2, it conveys that the cortical thinning, again an indicator of ALS progression, significantly correlates with the uptake of PET radioligand. So, and this was established between a comparison between the two cohorts, the patients with ALS and the healthy control individuals, HC. Finally, figure 3, the graphs shown above, 3 graphs, they demonstrate that correlations between standardized uptake value ratio within the precentral gyrus of the brain, bilateral precentral gyrus, also known as PCG, the fractional anistropy, FA, in bilateral precentral gyrus, PCG, and thickness of bilateral PCG. And all of this is correlated with the clinically determined measure of patient upper motor neuron burden, UMNB. So this correlation signifies that the presence of ALS is very significant. These are my works cited. And I was inspired to pick ALS as a disease because my beloved grandmother suffered from the disease for many years and passed away due to it. Thank you.